Important question from plant respiration. Question number 21. The number of substrate level phosphorylation in one term of citric acid cycle is 1. There is only one substrate level phosphorylation is there, in which directly GTP is produced and then this GTP is converted into ATP. This takes place during the conversion of succinyl coenzyme A to succinic acid. Question number 22. In the electron transport system present in the inner mitochondrial membrane, the complex of 1 and 4 are respectively, this is option number 3, that NADH dehydrogenase and cytochrome oxidase complex. Complex of 1 is NADH dehydrogenase, complex 2 consists of FADH2 and it contains FAD and iron sulfur protein. Complex 3 is cytochrome BC1 complex that consists of cytochrome B, cytochrome C and iron sulfur protein. Complex 4 consists of cytochrome C oxidase and this contains cytochrome A, cytochrome A3 and 2 copper center. Complex 5 is ATP synthase. Question number 23. The small protein attached to outer mitochondrial membrane which act as mobile carrier for transfer of electron between complex 3 and 4 is the cytochrome C. In this picture, this is the cytochrome C, the small protein which transfer electron from complex 3 to complex 4. Question 24. Cytochrome oxidase, that is cytochrome A3, contain option 3 both copper and iron. Other cytochromes, they contain only iron center, but the cytochrome oxidase, the cytochrome A3 contain copper in addition to iron for the transfer of electron to the oxygen. Iron pass the electron to the copper and then finally copper pass the electron to oxygen. Question 25. In cellular respiration, the final acceptor molecule of proton is the oxygen. Oxygen accepts the electron, the proton and make the water. This is known as metabolic water, and so oxygen is utilized at the end of the respiration to accept the electron and proton. Question 26. Oxidative phosphorylation is option 1, formation of ATP by energy released from electron removed during substrate oxidation. This is the correct definition of the oxidative phosphorylation. Phosphorylation is the production of ATP molecule, and for ATP synthesis, energy is required, and this energy here comes from the oxidation of the respiratory substrate. Question 27. Chemiosmotic theory of ATP synthesis in chloroplast and mitochondria is based on proton gradient. For ATP synthesis, a proton gradient is developed across the membrane. In case of mitochondria, it is across the mitochondrial inner membrane, and in case of chloroplast, it is across the membrane of the thylakoids of grana and the stroma. So when this proton gradient breaks, the energy is released and that energy is utilized for the synthesis of ATP in presence of enzyme ATP synthase. Question 28. The energy equivalent of a NADH is which of the following number of ATP molecule? There is option 2, that is 3 ATP molecules are produced from one molecule of NADH when it is oxidized. One molecule of NADH has enough energy to support the synthesis of 3 ATP molecule. Question 29. In aerobic respiration, total number of ATP molecule formed from one glucose molecule is option 3, 36 molecule. After the complete oxidation of one molecule of glucose, 36 ATP molecule are produced. 6 molecule from the glycolysis and 30 from the oxidative decarboxylation and Krebs cycle. Question number 30. This is based on the respiratory quotient, and this question is frequently asked. You can see from here that question 30A, respiratory quotient value of tripamidine is, it is option 3, that is 7, and 30B, respiratory quotient of glucose is 1. 30C, RQ of protein, carbohydrate, fats, and organic acid are in order of, this is option 1. Protein has less than 1, carbohydrate 1, and fat is less than 1, organic acid are more than 1. RQ value of the respiratory substrate depend on the relative quantity of oxygen present within the molecule. As protein and fats, they contain less oxygen in compared to carbohydrate. That's why protein and fat has RQ value less than 1. If we compare the protein and fat, then fat has a lesser oxygen than protein, and that's why fat has the RQ value 0.7, whereas the protein has RQ value nearly 0.9. And organic acid has very high oxygen, and that is why it has to absorb very, very less oxygen from the environment, and so its RQ value is greater than 1. Question 31. Aerobic respiratory pathway is appropriately termed as option 3, amphibolic. Though aerobic respiration, it is a catabolic process, but the intermediate metabolites that is produced during the Krebs cycle and glycolysis, they are utilized also for the synthesis of several important biomolecules, and in those cases it functions as a anabolic reaction, and so this is known as amphibolic, that is both catabolic as well as anabolic. Question 32. Select the wrong statement. Option 1. When tripamidine is used as a respiratory substrate in respiration, the RQ is 0.7. This is true. Option 2. The intermediate compound which link glycolysis with Krebs cycle is malic acid. This is wrong because this is acetyl coenzyme A. Acetyl coenzyme A, the correct one. So our wrong statement answer is option 2. Option 3, one molecule of glucose yield a net gain of 36 ATP molecule during aerobic respiration. This is true. Option 4, one glucose molecule yield a net gain of 2 ATP molecule during fermentation. True.